Hello, my name is Manuel Hernandez, and on behalf of my co-authors, I would like to present multimodal classification of Parkinson's disease using delayed differential analysis. First of all, I would like to thank my co-authors, Howard Poisoner, Terence Sanowski, Jonathan Weihenmeyer, and Claudia Lanschek, as well as collaborators at the University of California, San Diego. Parkinson's disease is a debilitating degenerative motor disorder that affects many older adults. Parkinson's disease affects nearly 1 million individuals in the United States and is characterized by a loss of dopamine-containing cells in the substantia nigra. The dopamine deprivation leads to nuclei within the basal ganglia to become dysfunctional. Motor symptoms in Parkinson's disease may be a result of markedly abnormal patterns of timing and synchronization within basal ganglia cortical circuits. Thus, cortical electroencephalographic signals recorded at the scalp can be used as a marker for the nature of the processing within these altered circuits. Previous work has shown that Parkinson's leads to alterations in the control of volitional eye and arm movements. In contrast to age-matched controls, PD patients more closely track their fingers with their eyes and, as can be seen here, while healthy controls typically will fixate on a lateral aspect of the target and maintain their gaze there in that position without having that synchronization between the movement of their fingers and their eyes. In this bird's eye view of the thumb and index trajectory, you can see that control subjects have more consistent movements, irrespective of the object or vision perturbation, whereas Parkinson's disease patients demonstrated more abrupt changes after an object perturbation. In addition, PD patients move slower with more pronounced arrest in speed. This can be seen by this dramatic increase in terms of the maximum velocity. Considering the trajectory of hand velocity over time, normalized from the tone onset to the grasp, we see this change after the object perturbation more clearly in Parkinson's disease patients, both on and off medication. Considering a 1-second sample of EEG data in a healthy older adult and PD patients off dopaminergic therapy, we may find it difficult to distinguish differences due to Parkinson's disease in a brief recording. However, by considering a delay embedding of the original time series, we're able to uncover some structure in the underlying time series data. Considering a delayed differential equation model consisting of both non-uniform time delays and a derivative of the original time series allows for a generic embedding of the data so as to capture additional information about the underlying dynamical system. In this paper, we aim to answer the following question. Can delayed differential analysis be used to differentiate between PD off dopaminergic medication, or PD off, and healthy age-matched control subjects using brief behavioral and EEG data? In this virtual grasping experiment, subjects sat with their thumb and index finger attached to a pair of phantom robots that allowed for high-precision motion capture and haptic feedback. In addition, subjects had their eye movements and cortical activity recorded using an eye tracker and a high-density 64-channel EEG system. Subjects rested their fingers in a virtual dock until an auditory cue was provided, which prompted them to grasp a virtual object at a comfortable pace. Sometimes the object was rotated during the movement, thus requiring a change in grasp configuration from a narrow to wide aperture. On other trials, visual feedback of the digits, presented as white spheres, was removed as soon as the fingers left the dock until reaching 2/3 of the reaching distance, so as to not be absent during the feed-forward control phase of the movement. In this study, we consider only successful grasping trials, meeting the accuracy criteria, which require the left and the right hand sides of the object to be grasped. Using a virtual reality environment allowed us to carry out precise vision and object perturbations in this 2x2 factorial design. In this study, we focus on full vision trials on a 750 millisecond period after object perturbation. Multimodal data included 64-channel EEG data, 2D gaze position, 3D hand position, average hand speed, sagittal hand velocity, and aperture, sampled or downsampled to 512 Hz. DDA, or delayed differential analysis, is carried out strictly in the time domain and not frequency domain. Considering a segment of EEG or behavioral time series data sampled at 512 Hz, we propose the use of a specific DDA model with 2 time delays, 2 to 3 terms with coefficients, and up to a third order nonlinearity for any given electrode or given behavioral time series, which relates the derivative of the time series data at a given time point to values of the function at previous dates. The DDA model consists of a linear term corresponding to dominant frequencies and timescales, and a nonlinear term corresponding to phase couplings and feedback. Now, considering the local region, about a single point in time, we use a 4-sample derivative margin before and after a given time to calculate a second-order derivative of the time series data. For any given trial, DDA is limited by the derivative margin and the maximum time delay, or tau max, constrained between 1 to 120 time steps in this analysis, which leaves us the following data segment for feature extraction. Given a time delay tau 1 and tau 2, we can step through the remaining data using a fixed window size of 1.5 second and 1/20th of a second step length to calculate the residual or least square error between the model and derivative and linear and nonlinear coefficients at a given time as features of the EEG and behavioral data that are then used for characterizations and classification. We can then apply a DDA model to a segment of data from a control subject to find its relative position within this hyperdimensional feature space. Running through the remaining control subjects, we would expect a cluster of outputs which should contrast with the output from PD patients and would allow for the calculation of a hyperplane using a singular value decomposition in red that provides the best separation between these two clusters. This then allows us to collapse the results of the model into a single dimension, which we will call the distance from the hyperplane. We implemented repeated random subsampling cross-validation. We used 2/3 of the data as a training set and the remaining 1/3 as a testing set, resulting in 84 training and test phases. The average weight matrix was then applied to remaining validation dataset. For multimodal classification, the top 20 model delay pairs were selected for each single modality DDA, resulting in 400 features used for each EEG behavior combination. DDE classification of EEG and behavioral data for perturbation trials resulted in better performance as evaluated by the area under the receiver operating characteristic curve, or AUC, in behavioral modalities, such as aperture during perturbation trials and velocity during no perturbation trials. When spread across the scalp, the combination of EEG and behavior appears to influence the topography of classification performance relative to the original EEG data. Improvements were found in multimodal DDA classification relative to unimodal classification, particularly in aperture during perturbation trials and velocity during no perturbation trials. Based on the histogram of top-performing model delay pairs, we find that EEG models appear to be task invariant, with similar linear terms for both trials, and small differences in the order of the nonlinear term. The top delay was 45 time steps, while differences in distribution in second delay suggest differences in the temporal scales that more robustly separate the two classes. The top-performing models and delays are significantly different across behavioral measures and also experimental paradigms, suggesting distinct dynamics and temporal scales in perturbation versus no perturbation trials. In conclusion, delay differential analysis can be used to differentiate between PD-off dopaminergic medication, or PD-off, and healthy age-matched control subjects using brief behavioral and EEG data. Multimodal DDA can be used to boost the performance of nonlinear classifiers in the differentiation of health from pathology. I'd like to finish by thanking my co-authors and collaborators, as well as the financial support from the Howard Hughes Medical Institute, National Science Foundation, and the Office of Naval Research, Science and Technology. Thank you very much for your attention.